Good afternoon, everyone. My name is Rashia Deepti Ghosh, and I'm assistant editor with Elets Technomedia. We are here for a panel discussion that is being hosted during Elets Diagnostic Leadership Summit. The topic of the panel discussion is leveraging clinical LCMS for advanced diagnostics. With us, we'll be joined today by Dr. Akhilesh Pandeyji. He's MD, PhD, Professor, Department of Laboratory Medicine and Pathology, Center for Individualized Medicine Clinic, founder and director, Institute of Bioinformatics. He'll be moderating the session for us. Other panelists who have been joined us, who are a part of this panel discussion are Dr. Prakash Srivatsa, who is DGM, Advanced Diagnostic, Apollo Diagnostics, Dr. Madhurika CH, founder director, Vijay Rekha Life Sciences.
So I think we lost our introduction, but perhaps we can go around the team and just introduce ourselves accordingly. On my screen, top left is Dr. Pandey. Perhaps Pandey, you can kickstart for us.
Yes, I'm Akhilesh Pandey, and I'm a pathologist by training, and I'm devoting a lot of my time to new assay development for clinical diagnostics, especially using mass spectrometry-based technologies. I'm physically at Mayo Clinic in Rochester, Minnesota, but I also founded a nonprofit systems biology institute called Institute of Bioinformatics in Bangalore, in India, where a lot of this work is also happening.
Hi, everyone. I'm Dr. Srivatsa. I head advanced diagnostics for Apollo Diagnostics, which is a part of the Apollo Hospitals group. And I look into more of the techno-commercial aspects of how we can make sure we leverage LCMS as a platform to ensure we give the best of results at the fastest possible time to our patients. Yes, I am a pathologist too by profession, but I do more of clinical commercial work. I'm based out of Hyderabad here in India.
Thought I finished. Hello, this is Dr. Madhurika. I'm basically a doctorate in biochemistry, being using mass spectrometries in different fields. We started Vijay Rekha Life Sciences to leverage LCMS tests into diagnostics. And we would like to see more and more tests come onto the LCMS for rapid, accurate detection of components and give accurate results to the patients. Thank you.
Good afternoon, good evening. I'm Dr. Punita Bhatia. I'm an MD in biochemistry, and I have been working in clinical diagnostics, clinical laboratories for over 21, 22 years. My experience with the LCMS has primarily been in the fields of TDM in transplant patients and in newborn screening disorders. Looking forward to be a part of this very inspiring discussion today. Thank you.
Good morning, good afternoon, good evening, everyone. My name is Dr. Edward Gaucher. I've been working with Thermofisher Scientific since 2010, and I report into the chromatography mass spectrometry division, where we manufacture the mass spectrometry products for the clinical market. And a big part of my role is really focused on market development activities. Lots of background noise. That's better. So a big focus of my role is aligned to market development activities, working with our customers, with our collaborators, our key opinion leaders to understand market requirements, future trends, to help us as a manufacturer develop, research, and commercialize the best suited instruments ready for the clinical market needs.
Hi, good evening. I'm Dr. Nilesh Shah. I'm a president of Metropolis. I've spent 35 years in the company. Metropolis is a reference chain of labs in the country, 4-decade-old company known for starting a lot of special tests. We also have mass spectrophotometry, and I'm going to share some of the struggles and positive outcomes which we had ultimately. And I'm very happy to be part of this forum and contribute. Thank you.
Dr. Kansal? You are muted.
I think we can start, and when her turn comes, she can introduce and
Yes.
answer.
Yeah, sure.
Sure.
So I think I gave a brief talk, but many of you are practitioners of the field, and in India, you have developed tests, you have deployed tests. I think it'll be a great way to start to see where LCMS-based tests are in both newborn screening, inborn errors of metabolism, and others as we go forward. So I think we don't have any particular format for this, and we are still waiting for one person who is muted now and one more to join. But we can start with Dr. Shah. You said that you have had some experiences, and it would be good to hear about your experiences as you've deployed some of these technologies.
So I come from a very pure clinical diagnostics area, less on the research side. And as a reference lab, when we started, our start journey itself started with immunoassay. So we started as a thyroid testing lab, thyroid and fertility testing we used to do early days. We never used to do routine. And we have been more used to doing tests which are automated. From 90s when chemiluminescence started and some sort of automation started, we have been used to that kind of a journey. So the companies who used to supply us, support us, technically equip us, they were also tuned to that. About 12-15 years back, when mass spectrophotometry was talked about and entered clinical diagnostic market and reference labs, we were also one of the adopters. But we went through some struggle, and there have been some good outcomes as well. So I want to share that in my coming 3-4 minutes time. So if I tell you the initial first biggest struggle which we faced is this was not as easy as buying an equipment or immunoassay equipment and loading a kit, calibrating and running the test. You needed much better talent, though I have all my life done that immunoassays, but it needed much different way, mindset, talent, and patience to start any assay. And this is the first technology which we didn't go live for 2 years. 2 years we kept on struggling with people, assay development, getting quality assured. Okay, I think quality is good to give, and so on. We were confused at every level. How to even deal with the companies, whether to buy Thermofisher equipment, Waters equipment, or anything else. I think the struggle started from there. Even commercial deal, if I talk about to some extent, the kind of capital investment which it incurred, it was hard for us to digest, because otherwise we get machines at 1/8, 1/10 the cost, and at times rental. Here it was not the same. The whole deal making with the supplying company itself was very different. One day, after a lot of struggle, we made a decision. We got it, but it took time to get that kind of talent and start the test. Uh, During our 2-3 years journey, we also had a lot of confusions, and I don't want to blame the supplying company every time. I think problem was at our end as well, where maintenance of the machine, spare parts of the machine, the way it used to happen with immunoassay companies, or chemistry companies did not happen here. In analytical chemistry, we felt it was very different ball game altogether, and at times we lost patience also. But overall, when we started with TDMs and the quality of results we started getting when we were set, I think we were pleasantly surprised. Pleasantly surprised to a level that we really felt that something which is gold standard is with us, and hence we became very keen on growing this technology for many more assays and ensuring that it becomes part of our menu and it gets promoted. Uh, we started with TDM and NBS like most of the people do it, but very keen to have something more like testosterone assays, I think. And I've spoken to a lot of gynecologists, I've spoken to a lot of urologists who use testosterone assay or free testosterone assay, and right from reference ranges to the correlation with the clinical findings has not been to their satisfaction. Testosterone assay, which gives you full testosterone, which includes your diodotestosterone and SHBG and everything, I think I felt it's very popular. That is what we are right now at developing stage. In clinical diagnostic, I would also lots rely on LCMS for catecholamines, metanephrines, many of the vitamins, amino acids for sure. I think is a very good assay for, and a lot of steroids. This has been my wish list, which some we have already developed and rolled out, and some we are on the way of developing. This is one area which I want to talk about. And when we talk about MS, I think I should also talk about ICP-MS, where a lot of heavy metals, since we gained some experience, we have done it and we are very happy with it. It has been well accepted, and with clinical sample, doing any heavy metal analysis was not easy for us. When the company was not actually analytical chemistry strong company, it has been difficult, right from setting up reference range, validating ranges, and everything has been hard. But once it was done, I think we are very happy with the result. Clinicians are happy, and whenever we have discussed any particular cases, I think our result was appreciated. The something which we don't have today is a GCMS, which we don't have, and I'm looking forward to it in the future to come. One part I just want to indicate along with all this is somehow we are still not set to do very large volumes on MS, like in absence of automation. Like we are used to an automated chemistry equipment that you load the sample, it will read the barcode, it will pipette automatically, and straight away throw the result, and result will go back to your through interface. It will go on to your LIMS, which does not happen with mass spectrophotometer. This is going to be a journey, and hence any assay where we have large volume, we are still little bit staying away from MS, and we have to depend on MS, our regular immunoassay system. So this has been my small little experience on MS. I thought I'll contribute for the audience, and over to you, Dr. Pandey. If any more thing I can answer, I'll be happy. Thank you.
Thank you so much for sharing. I would like to continue before we get to a little more discussion points. I think I would like to continue in the same vein and ask Dr. Madhurika to tell us about some of the challenges that are faced by new diagnostic labs in India and how they are navigating the landscape.
I've been associated with Tenet Diagnostics in Hyderabad, and I have set up the lab there initially, and with Sandor Life Sciences. We started up Vijay Rekha, looking at the challenges that the diagnostic centers face, especially in the normal treadmill diagnostics that happen. As Dr. Nilesh has expressed, it is very difficult. The mindset has to be different to analyze, accept, and also put the test and make it a routine test, even if the numbers are more. So basically, you should understand how LCMS is working. You should understand what are the instrument parameters that are vital for giving the results. It also it is the best thing that can happen for all biochemical analysis. But these are the points in India. First is the expertise, as Dr. Nilesh has said, it's very difficult to find people who have expertise in running the instrument. It is not that it cannot be run, because triple cord quantitative analysis is very well established in biopharmaceutical industry, and they are well trained manpower. But when it comes to diagnostic and to get those people onto the board to give the TAT and to reach the accuracies consistently on the instrument. These are the challenges that any new or experienced players of diagnostic companies are facing today in India.
Thank you for sharing.
Yeah. I hope I answered your question.
Yes. Yeah. So I think I would like to revert back to Dr. Gaucher and ask him to comment on some of the issues that were brought up, especially one is assay development, but also about doing this on large scale. Dr. Shah said at the end, he's very happy, but he wishes he could do more samples. It is a great technology, and perhaps Ed could talk about automation and things like that as they would apply to a very rapidly evolving market in India, one would say.
Yeah, sure thing. And thank you for the handover. So something that keeps on resonating in my ears when I was listening to these discussions was that the mass spectrometry is still seen as a very much a focus of research instruments. You need a PhD to run it. You need technical ability. And as a manufacturer, this is something that we're very much aware of, and we've been working very hard to resolve these requirements and make it easier for clinical scientists, for people without mass spectrometry or without much analytical experience, to run these instruments. And we've come a long way. I guess it's not just the hardware, it's also the software that is a big driver, and then the method setup and development. So at Thermo Fisher Scientific, we over the years have been pulling our capabilities from improving sensitivity of our instruments, but through to automation and sample handling. Now, there's two parts of this story. One, the full analyzer route, which would be in the core laboratory, and mass spectrometry as it is, open platforms, is positioned in typically the specialized diagnostics facilities. And this really harnesses the power of mass spectrometry, the ability to add and remove your compounds. Anything that you want to analyze, you can develop an LDT, run an IVD assay. So you really have a lot of flexibility, a lot of capabilities with mass spectrometry. And it's those capabilities that come with the ability to develop your own methods as well. So no longer do we just want to look at one or two compounds, you can look at panels. But with that freedom comes the challenges then of optimizing the method, the LCMS parameters. And as a manufacturer, we've been trying to improve that with innovative and streamlined softwares that improve the user experience. The instruments themselves, we're now looking towards improving the sample collection strategies so that we can automate the approach as much as possible. Now, with open platforms, we have a lot of flexibility. We have the ability to add additional configurations into the front of the mass spec. Dr. Pandey mentions the VeriSpray source. We've also recently launched a dried spot autosampler called the Transcend DSX1. This takes your dried blood spots or dried spot samples, so it could be urine, plasma, anything that absorbs onto a substrate. You can take these and you can automate the entire approach from as soon as the collection card goes into the autosampler right through to the top of the mass spectrometer. It has online sample preparation as part of it. So in the open platforms, that's one side of the story. The other side of the story is, as a manufacturer, we're also very much involved in mass spectrometry as an analyzer. And some of you might be aware of the Cascadian project. You might have heard of it. I must say that it's not currently available in India, so it's available in select countries at the moment, select European countries and North America. But I want to mention it because it really does start to identify the way that we're going in the IVD space. This is mass spectrometry in a box. This means that we have the ability to run the system as an analyzer, where it's essentially random access. You put in a blood tube and it will run for select diagnostic tests. And this means minimal user input, minimal training required. And it's the approach towards full automation. And this is then positioned in the core laboratory. So you have the complementary availability of an analyzer with mass spectrometry. And for those that want to continue in terms of open platforms with the ability to add and remove, or if the menu isn't ready on the analyzer, you have the opportunity to use the open platforms as well. And this meets the needs then of all technical abilities in the laboratory. And essentially what we do with our open platforms and what we're doing with the developments on automation, dry spot analysis, VeriSpray, we look to the future and how can that go into an analyzer system as well. I also heard some comments around volumes. This is a key part. With analyzers, you typically need a volume of test for it to be economical in terms of cost per reportable results. And of course, this is something that we always discuss with our customers. We have financial solutions in place to make sure that this is appropriate. And the nice thing is the portfolio on our open platforms can be scaled up. So you can have a portfolio that is suitable for a small volume of samples or large volumes by increasing the sample capabilities, the sample throughput. So we can address all of those challenges. Okay, thank you.
So I have a question. The dried blood spot order sampler you're talking about, is that being developed or is it already operationalized and available?
It's operational and available. We commercialized and launched during ASMS last year. Okay. We're very excited about this project. We've been working on it for a long time. And essentially it works by flow through desorption. So a clamp positions over the dried spot, a flow through desorption into the line of the LC or into the mass spectrometer directly. And really what differentiates this technology is the ability to do online sample preparation utilizing the TurboFlow technology. And this gives us tremendous advantages to remove matrix interferences and optimize sensitivity and of course selectivity. It works across all of our mass spectrometry platforms. They are, in this case, the detectors, and it's unified in one software. So please, if anyone has any information requests, let me know. I'm very excited about this product, and we're very keen to discuss it further.
Yeah, thank you.
So this is attached only to the, sorry.
Go ahead.
This platform is attached only to the triple quad or also the intact mass for high resolution?
Across all of our mass spectrometers. So from triple quadrupole to Orbitrap to Iron Trap, anything that runs under a specific software that our platforms operate. So we have a lot of flexibility.
What is the capacity of sample load?
I believe the autosampler holds 86 cards on this first generation. And each card has 4 or 5 spots, depends on the type of card that you put into it. Whatman, PerkinElmer, Himaxis cards. So it's filter paper. Some people are using cellulose. Other people are interested to look at other substrates. As long as it fits within the clamp that we position, we have a lot of flexibility.
I see that Dr. Seema Kapoor has joined. It will be great if you can give a brief introduction because the rest of us have already introduced ourselves. Are you on mute? Can you unmute? No, you are still on mute. So, yeah, okay. I think, oops,
Nope, we lost.
we lost it. Okay. Maybe we'll wait for her to come, but this is a question for Dr. Srivatsa. How are the regulatory compliance aspects pertaining to, say, CAP accreditation adopted in the Indian diagnostic market? And also, if you can comment if there are more labs in India adopting the NABL or CAP accreditation when it comes to testing for human samples.
Yeah, hi. Thanks, Dr. Pandey, for the handover. Yes, CAP has been a very, I mean, coming from a CAP laboratory myself, Apollo Diagnostics, they have been very rigorously questioning us on how we have been developing our assays, especially if they are home brew and they're laboratory developed tests. So that's a very important aspect I think we need to look into. But to facilitate high-end tests like LCMS, GCMS, and Dr. Shah mentioned ICP-MS, to become accredited with either CAP or NABL, I think what is mandatory is we all develop a global repository of data and start sharing, like what we do for genomics. For example, you have gene banks. So why can't we share our experiences on a common platform, if that makes sense, so as to ensure we learn from our mistakes? And the more these tests are accredited, and as we all know, people like CAP, and if they are going to validate your platform, and then if they say, hey, you're good to go, it makes a lot of sense from a business standpoint and also from a quality standpoint. I'd love to hear what others would have to say, and most importantly, you from from Mayo. But yeah, more and more labs want to go for NABL and CAP because our patients are also becoming very aware of regulatory protocols that exist in the country for healthcare services, right? So how are we going to make sure that we give them that quality? Yes, Dr. Shah, I think I'm sure he'll have something very interesting to say.
I just want to share some experience on it. So, many a times you look forward to some appreciation coming from clinician. If I'm giving tacrolimus level on a regular immunoassay machine and giving on LCMS, I've taken different level of pain, and my quality also LCMS is gold standard. But barring few one or two tests and barring few selected clinicians, they do not understand whether you are done by regular chemistry on ELISA or done on a good immunoassay system or on LCMS. This is something needs to be tried. This is one thing I just thought must indicate. And I think all people who are fond of this technology, I think all of us should do that. Let it come very prominently on the report, and whenever we are speaking, we should talk about it. Second aspect, what you raised, Dr. Srivastava, very good aspect that, when you develop an assay and you have run it, you also want to build a confidence, because your confidence will come out when you're talking to a patient in case of controversy or in case with a doctor. If you have done a proficiency testing with something like CAP and your results are consistently passing, you are sure about it. And this is important. And I want to give you one example. And pardon me, I just don't want to call names, but this is about 8, 9, 10 years old story. When we used to do tacrolimus on architect system, the immunoassay system, and there were some 7, 8 labs who have been doing on LCMS. And one of the pharma company took a lead because they have been getting a lot of feedback from clinicians about results not being good. So one of the pharma company got 4 clinicians together, 4 leading clinicians of Bombay. All of them gave split samples to various labs, and one sample was going to a UK lab who is like considered to be gold standard on proficiency testing for this. And this journey was 3 months. 3 months continuously they did that, and afterwards one day we were all called to see the result. We were not even knowing this is happening. We were suddenly called without any agenda. You have to come and meet us. Now that pharma company happens to be our customer, so all of us went. And when we sat in the room, 4 clinicians started firing all of us, everyone. Okay, you people give wrong report, because of which we are having rejections. And that gentleman who came from UK, he presented the result. You would be surprised the kind of difference in results which were seen on tacrolimus from lab to lab was enormous. I don't want to say that we have been best, but our results somehow matched very well, and it was appreciated. We had done on immunoassay. We didn't have LCMS at that time. We had not standardized 10 years back. I'm saying it was correlating well. But why I'm raising this point, not to say that Metropolis has done good, but I'm saying the standardization on LCMS is so critical that if you do not do it well and if you do not have a good proficiency testing, the way Dr. Srivatsa said, you may go wrong and you may not realize. These are all good labs. They have a good track record, but somehow on tacrolimus, they did not pass the test. So having an accreditation and doing this good proficiency testing is critically important. Thank you.
Thank you. I think it's a very good point. Yeah. And I also liked Dr. Srivatsa's point about sharing of data, global repository. And I think we are not there yet. Perhaps we could develop a good ecosystem, and maybe our industrial partners like Thermofisher can help us do that. So, because I think unfortunately, there is no direct connection across labs. That's one of the unfortunate parts, but we do rely on hardware for companies. And I think companies like Thermo Fisher then perhaps could be innovative and create those platforms for us to do that. And of course they win, but I think standardization, I would say, definitely I agree with this, that it is an issue. Just because it is a promising technology does not mean that it does not have any of the evils of any technology which is in the early phases. So Dr. Siva Kapoor has just joined again, so I would ask her to introduce herself and then we'll continue our discussion.
Yeah, I'm extremely sorry for joining in late, and I'm a director professor of pediatrics and entered into the mass spectrometry arena about around 15 years from today to look at expanded newborn screening, because my primary interest has been newborn screening. And now that we know that many of the disorders which were thought to be non-amenable to mass spec screening, even the common analytes, can easily be standardized on the mass spec, though we definitely share the issues that sensitization, critical handling of samples, and standardization of the LCMS platform with good quality assurance remain challenges for labs which are not headed by biochemists. And when you start entering into an arena, which is little naive to what you've done. But yes, like diarrhea dehydration is very, very interesting to the pediatrician, so is metabolomics. And that's how I entered into this field of LCMS. That's a very brief introduction that I would like to do.
Yes, and I would like to follow, because you're a pediatrician, I would like you to zoom out a little bit and tell us about what the awareness for newborn screening is in India, what is being done and what can be done more for it to basically make it a routine requirement. And as you know, in many of the Western countries, it is a regulatory requirement, and that's not the case in India. So we'd love to hear your thoughts on that.
Yeah, so Dr. Akhilesh, I would just like to say that today in Noida, the Pericon is going on, and Pericon is the annual conference of the Pediatric Complete Association, and tomorrow I'm going to speak on newborn screening as a plenary session in Pericon. So the awareness of newborn screening has really jumped a lot over the last 10 years. Since the ICMR started its first project in the year 2004, followed closely by Science and Engineering Research Board in the year 2014, and now we have recognized, and we've just got through a project again from DHR, which looks at first tier newborn screening on the expanded method, as well as second tier newborn screening using a combination of genomic and metabolomic technologies. So this final omics is definitely going to gain sight, and probably it's not going to walk, it's going to jump in India, because the pathway and the awareness has been created. And of course, we have all sessions going on which look at newborn screening should be mandated as a law for the country without doubt for certain panel of core disorders, and amongst them LCMS should be made a must for all babies getting admitted to SNCUs, that is sick care newborn units, and neonatal intensive care units of all levels, because these babies will not then receive screening but actually a diagnosis once they go into the era. And now that the wedding between genomic scientists and the metabolomic people have occurred, I think there's nothing better to complement omics in a complete way in newborn screening. So I would say that currently there are about 20 to 25 labs in the country which are looking at metabolomics, and much more than that in genomics. So what is required is a good wedlock between these two disciplines. And yes, I definitely agree that we should be sharing our data, entering into a good quality control program, because the positive predictive value of most analytes is between 35 to 50%. Adding ratios, second tier screening would make it improve by another 20%. Adding complementary genomics to the dried blood spot is going to actually make a big way. So maybe as IT has done, maybe bioinformatics, genomics, and metabolomics is going to be sky high in the coming decade in the country.
So how do you think technologies that you said like LCMS are very important, how can they be put more into use?
Yeah, so
Even though you
what-
said there are 25 or so labs, it's still inadequate. So how can this base be expanded to serve a big country like India?
Yeah, so I think there are a few crucial things that we have to have in place before we expand the setting that we have. One is that the diets have become available, so screening by ethos is not non-scientific, because earlier when we started and we submitted a project, we were told to change our title from newborn screening to data generation for non-treatable disorders. So today we have diets which are in house, but we are still somewhere lacking in the emergency drugs needed for newborn screening. What we require is partners for making these very, very economical drugs in the country. And all pharmaceuticals in the country should look at a disorder which can be as common as 1 in 1200. And if you look at the birth cohort, I think you would be saving one baby every hour in the country if you were to actually have expanded newborn screening. But before the bottlenecks are met with, the process will be a little slow. Once they are all in tune, we can go expanded in sick care newborn units using LCMS as a platform, not only for IEMs but for thyroid, for hemoglobinopathies, and many other things that we don't even think about as point of care tests on the blood spot, which can be added in the newborn screening paradigm.
Yeah, I liked your comment about hemoglobinopathies. And I don't know if you listened to the talk that I gave. I think we are developing an LCMS-based platform that can actually do just that, because I think it should be considered together with the other disorders. Thank you for your comments. I have a question for Dr. Bhadia, if she can share her experience uh, uh, on newborn screening and the different
Yeah.
kinds of assays.
Hi. So it's been very nice listening to everybody's views. I would concur with Dr. Seema Kapoor and Dr. Nilesh Shah. The first thing I feel where we lack in India is awareness. Having worked across both reference labs and in hospitals, I find there is a very niche set of physicians or pediatricians who want us to provide newborn screening. My association with newborn screening goes back about to 2008 when I joined a hospital in Bangalore, where I was working for about 5 years in that hospital. Our neonatologist had just returned from the UK, and he was well trained and he was aware, and he said that when we go live in this hospital, you will provide newborn screening to every baby born in this hospital. That was the driving force. But across the chain of the hospitals, he was probably one of the few pediatricians who felt so. But with his target, initially we were outsourcing it, and slowly we internalized it. We started offering a basic panel of 4 or 5 assays. Again, so the in India there are, I don't know if I'm right, Dr. Kapoor, please intervene, Dr. Nimi. So there's a basic panel which is more competitively priced, which consists of TSH, biotinidase, 17-OHP, galactosemia, and cystic fibrosis and phenylketonuria. These are usually done as a basic screening panel, and that is offered to most babies, normal babies who are born, as a screening. And the LCMS is usually reserved for the sick babies. But whereas abroad it is part of regulatory, I think we are far from that, where we are actually giving it to all the babies born. It is selectively offered to sick babies. That also, even the hospital will decide very late to advise the LCMS testing or tandem. They call it tandem mass, whatever it is, whether it's GCMS or LCMS. It's very late, and the baby is usually very, very critical by that time. As Dr. Seema said, it should be a part of the protocol for metabolic evaluation for every sick baby or infant who is admitted. Why even babies? Children who are presenting to neurology and other developmental OPDs with delayed milestones, they should also have a mandatory screening. In fact, when I was standardizing LCMS for a previous organization, we had difficulty in finding reference ranges. Why do we limit our reference ranges only to babies, whereas anybody with retarded milestones or development can present at any point in time to a hospital? At NIMHANS, that's the Institute of Behavioral and Mental Health in Bangalore, they've done some good work there, because the biochemist leads the cell there, and they've got some good papers. I'm sure Dr. Kapoor would know. So that is my first thing, is that the awareness, and even clinicians don't insist. They don't insist on. So it'll come into more into importance, more into work if the clinicians again want it. Same goes for the transplants, as you said. I have worked in organizations where we were offering tacrolimus, cyclosporin, everolimus, sirolimus by tandem mass spec. Again, it was the clinician, the transplant team, which valued these reports. Sadly so, we didn't find a suitable EQUAS program in India. We were taking an RCPA from Royal College of Pathologists, Australia. We are using the EQUAS from there. So that's again one of the, although it's being done across the country in so many labs, but we don't have a EQUAS program. Similarly, even though for newborn screening, it was CAP accredited, but we don't have a EQUAS program. We were using the CDC program. So EQUAS is also EQUAS PT. This aspect also needs to be developed very much in our country to make it, again, even for accreditation, all this is important to close the loop. So I think the first step is awareness, the requirement, and at least building this into panels of at least corporate hospitals or people who can afford it. That is still not happening. That even the best of hospitals, when they are paying lakhs of rupees, let's say even for a normal delivery or for a cesarean section, 3,000 or 4,000 for a tandem mass is not being incorporated in their package. I personally feel that this is where we can start. The way I started off with NBS on a small level, because the pediatrician was keen that every baby would deliver here will go home with an NBS report. Similarly, I would request pediatricians to, kind of put a pressure on us or motivate us to imbibe this in our protocols.
Thank you. I think this is an important part, perhaps not widely advertised, because either we think of the clinical side, only the lab side, but the whole loop is not completed until there's awareness. If the physician doesn't understand the importance, and it is still a long ways from being mandated. Maybe we are closer, as Dr. Kapoor is saying, but we are still not there yet. So we are relying on the pediatricians being highly aware, and then honestly, LCMS, but also mass spectrometry is a new technology, even for the specialists. Then we cannot expect pediatricians to really understand and know all of the nuances, but the awareness is definitely a key part. I would like to go back to Dr. Gaucher and ask him to comment on two specific things. One is a point that was brought up by Dr. Shah, for example, that the assay development took so long. What is it that companies like Thermofisher can do to make it easier for the next lab and the lab after that and others to actually deploy it? We know that there are issues about recruitment and people, but still the assay is not easy enough, is what I'm hearing. And the other is, what can he comment about medical device registrations and global regulation from his perspective?
Thank you very much for the questions. So you raise a good point in terms of assay implementation. And this really depends on whether it's a lab developed test and depends on how many compounds you're looking for, or if it's an IVD based assay. And that can really be focused on the medical devices, which I'll talk about in the second part of the question or my answer. But for lab developed tests, typically, the application universe that we as a team have visibility on is over 800 applications. So as you can imagine, the clinical market, from research through to toxicology, inborn errors of metabolism, the scope is tremendous. And we have a responsibility to ensure that our customers have the most streamlined experience of implementing those methods. So over the years, what we do is we work with our customers, with our collaborators, to ensure that the method parameters are suitable for the source conditions on the mass spectrometer. Because it's one thing developing a method, creating a technical note and sending it to a customer and say, hey, use this, go and validate. We need to make sure that it works. We need to make sure that it has utility, that we know that it works on our instrument parameters. So we test our instruments before they go out with standardized methods, immunosuppressants for robustness, steroids for sensitivity. And we have legacy data of 10 years worth of work on these instruments. So we know that these are great criteria to check that the instruments that we're launching are suitable for the clinical market. And then once that has been accomplished, for new methods, for lab developed tests, we work with our partners to ensure that these assays meet the technical demands. And then we develop technical notes to support the LCMS parameters. Now, of course, if it's an LDT, a lab developed test where we haven't done it before, or it's a different panel, because of course the panels can change, we work with our global support teams to ensure that we can support you on site and of course connect you with partners as well if it's challenging. From the medical device perspective, so as a manufacturer, we have responsibility to align with global regulations and requirements. We've registered our open platforms in over 26 countries globally. The bulk of those registrations, as you can imagine, have been in Europe because of the upcoming revision on the IVD directive called IVDR. We have registered in India the triple quadrupole portfolio called the Quantis MD and Altis MD. And we also have a front end called the Vanquish MD. And these instruments have been registered and listed as medical devices. They have the same performance as the non-MD version. It does exactly the same in terms of method performance parameters in instrument setup. However, it meets the future requirements of the laboratory. The instruments are manufactured and aligned to certain quality agreements to ensure that they are meeting all the local healthcare requirements. And in terms of support for these systems, we have a responsibility to make sure that your methods are up and running and that you can validate accordingly. So we make sure that we work closely with third-party kit manufacturers to validate, so that they validate their kits for use on our technology. Our instruments are Class A or Class 1, depending where in the world you are. The kits from these manufacturers have the intended use. They're the Class B or Class 2 kits, which hold the IVD status. So what we do is we make sure that those kits work on our systems, and we position our instruments with these manufacturers or with customers to do this. And then we develop the technical notes to show the performance of the LCMS assay. And this ensures that when you purchase a system, when you set a system up, you know it's going to work on your instrument because it's been tried and tested, and this kit has been validated on that technology. And of course, it's not the hardware, it's not the software, but it's also our support and service which play a massive role here. And we have dedicated certified support teams with this in mind.
Thank you for a great summary. Yes, obviously. And we have just about a minute or so left. I would like to ask if any of the other panelists want to bring up any other important point. One question that comes up is GCMS versus LCMS. And I would say that this, some of it is legacy. Many of the assays that were initially developed on LCMS, GCMS platforms can be developed and deployed on LCMS. They are perhaps even more sensitive. They can be multiplexed. So I think it's a very complicated question, especially I think in the Indian scenario, where sometimes it's considered confirmatory, sometimes it's considered screening, sometimes there are cost issues involved. But really, if I'm not mistaken, I think it was Dr. Bhatia who made the point that we need to be doing the confirmatory tests, really, because they are available, and in the other parts of the country, for the most part, we are seeing that those confirmatory tests are essentially done. And as we move forward on these LCMS and other platforms, I would like us to be able to think of them as the only types of tests that are available. And this is also complicated by the fact that the quality varies across the different labs. So I think we need to shape up our quality and go for the more definitive goal of a definitive identification through confirmatory tests. And many of them will fall in the LCMS domain, but really, as we all discussed, the platform doesn't matter, but we need the reliability and robustness. And I think I would like to end this session there, and thank you all for your very active participation and very critical insights on this very important topic.
Thank you so much, Dr. Akhilesh Pandey, for moderating the session so nicely. And I thank all the panelists for their insightful views on leveraging clinical LCMS for advanced diagnostics. Thank you, everyone. We would like to inform our esteemed gatherings about our upcoming events, which are the 4th Pharma Leadership Summit on 26th and 27th of April, the 3rd Radiology and Imaging Summit on the 26th and 27th of May, and the Medical Education Summit on the 23rd and 24th of June. We look forward for your constant support in future. I once again extend my warm gratitude to all our esteemed panelists for joining in, and please accept a small token of gratitude from our end through these certificates that we present to Dr. Akhilesh Pandey. Thank you, sir, for wonderfully moderating the session. Dr. Edward Gaucher, for your thoughtful views, sir. Thank you so much. Dr. Nilesh Shah, for his views. Thank you, sir. Dr. Madhurika, for your insightful points on the discussion. Thank you so much, ma'am. Dr. Srivatsa, for your thoughts on the panel discussion. Thank you so much, sir. Dr. Punita Bhatia, for your keen views on the session. Thank you, ma'am. And Dr. Seema Kapoor for her insightful views. Thank you so much. It was wonderful having all of you. Thank you.
Thank you. Thank you.
Thank you all.
Thank you so much.
Thank you all.
Bye.
Goodbye.